My name is Rogal, and before I begin, I would like to really thank the organizers for giving me the opportunity to speak here today. I am currently a postdoc in Anna Herland's lab in Stockholm, working on neural in vitro systems, but today I'll present part of my PhD work, which was on adipose on-chip systems and together with the microorganol lab in Tübingen. Before we'll jump into the topic of adipose on-chip, let's briefly cover the basics of why adipose tissue research is important. So first of all, we have a lot of it. Adipose tissue comprises about 20 to 25% of a healthy human adult's body mass, but can be up to 50% in obese patients. There are three different types of adipocyte, the main cell type in adipose tissue, namely white, beige, and brown adipocytes, which are each morphologically and functionally distinct from one another, but still share one common, very important function, which is that they are master regulators of systemic energy balance. Therefore, having an abnormal amount or function of adipose tissue predisposes to developing really a plethora of comorbidities, such as metabolic disease, cardiovascular disease, reproductive dysfunction, cancer, and it even affects the susceptibility to contracting viral or bacterial infections. However, traditionally, adipose tissue research has faced a couple of challenges. Concerning non-human studies, a translation of findings to human physiology is very often not possible due to severe discrepancies in the mechanisms between the species, especially when it comes to immunology and metabolism. Concerning human-centered research, you're either restricted to observational studies if you conduct in vivo studies, or if you conduct in vitro studies, your research might be hampered by challenging culturability of the adipose cells. So we see that there's an urgent need for physiological adipose tissue model systems, and one technology that could address these challenges is the organ-on-chip technology. The prevalent type of adipose tissue in our body is a white adipose tissue, so we primarily focus on this type of tissue. And since a physiologically relevant model should be capable of reflecting key morphological and functional features of the tissue in vivo, it's really crucial that we become clear about those aspects before starting to build the model. So this is what we'll do now. I think that you all know one of the key functions of white adipocytes, which is their storage and energy distribution function. So I won't go into detail here, but I would like you to appreciate that through their lipid storage functions, there are some challenges that arise if you work with mature white adipocytes in vitro. So they are very large and extremely fragile. They are buoyant, and so far there are no decent protocols for cryopreservation yet. Another critical task of white adipocytes is the secretion of signaling molecules. Having a look at the morphology of white adipose tissue, we have the adipocytes, of course, and they are surrounded by an extracellular matrix. But there are also other adipose-associated cell types, such as endothelial cells, adipose progenitor cells and stem cells, as well as tissue-resident immune cells, summed together as stromovascular fraction. And there are some circulating immune cells. We integrated all of these cellular components in an autologous manner in order to enable patient-specific models. And to obtain all of these different cell types and, importantly, to meet their individual requirements concerning in vitro culture, we first had to establish different handling and isolation procedures. In a nutshell, the white adipocytes, stromovascular cells such as tissue-resident immune cells, and endothelial cells were isolated from a skin biopsy with subcutaneous PET. For experiments involving circulating immune cells, we isolated circulating monocytes or T cells from PBMCs, which in turn were isolated from blood samples from the same donors. Of course, integrating all of these different cell types into one model will yield a quite complex model. So we proposed a strategy of a modular and flexible model build-up, which allows for some modularity regarding the integrated cellular components, so that you can tailor your model to fit a specific purpose. So what you can see there is a model in the side view. Let me get the laser. So the mature adipocytes together with the stromovascular cells, are injected into a tissue chamber compartment and encapsulated in a hydrogel. And this tissue chamber compartment is separated from a continuously perfused media channel, which can be lined with endothelial cells. And circulating immune cells can be perfused with the medium. Now let's have a closer look at this microfluidic platform. So the platform is made from two microstructured layers of PDMS that sandwich a porous PET membrane. And to the bottom, the microstructures are encased by glass slide to enable easy access for imaging. Design-wise, the features of the device are particularly favorable when working with these fragile cells, because first, the design of the tissue compartment enables a sequential injection of the individual tissue chambers. And second, due to the compartmentalization with the porous membrane, the tissues down here in the tissue compartment are entirely protected from any shear forces. Now, let's inject some tissues. So the white adipocytes are the main cell type in white adipose tissue, and therefore we ran a set of experiments directed towards the applicability and suitability of our platform to specifically accommodate white adipocytes. And I will share some of the key findings of this set of experiments now. So first we had a look at their morphology and found that the on-chip adipocytes were morphologically very similar to adipocytes from donor-specific explants. They were distributed in 3D throughout the entire tissue chamber, and we found them to be unilocular and covered with perilipin A patches, which are two very important hallmarks of adipocyte maturity. Concerning their functionality, we ran a couple of experiments or different versions of a real-time monitoring of fatty acid trafficking. So, for example, we could show that the on-chip adipocytes took up fluorescently tagged fatty acids of different length. We had a look at their long-term functionality, so what you can see here are fatty acids being taken up on day 36 of on-chip culture. And we could also simulate the effects of different glucose concentrations on our fatty acid uptake. After this evaluation of baseline functionality, we further looked into how the on-chip adipocytes responded to external stimuli. So, for example, by administering the drug isoproterenol, we could show that the uptake and release rates of the fatty acids were altered, and also we found that the glycerol release by the adipocytes was altered in response to different doses of isoproterenol. We further characterize our platform for establishing endothelial-like layers in our media channel. For time's sake, I won't go into detail in these results here because I think now it's time that we increase the complexity. And you might remember that we want to do so in a modular mix and match toolbox kind of way. So let's have a look at the other model options that result now. Adding another cellular component to our monoculture setups will yield the following co-culture system: adipocyte endothelial cell, adipocyte SVF, and adipocyte macrophage co-cultures. Adding yet another cellular component will yield the adipocyte SVF endothelial cell multiculture system. And by perfusing immune cells, we'll reach the final stage of the complexity. It's important that we keep in mind that by increasing complexity, we do increase physiological relevance, but at the same time, we also increase expense and effort. So how do these other adipose-associated cell types contribute to adipose tissue function anyways? To address this question, we ran a set of experiments comparing these different model options regarding different Watt functions. And for maximum comparability, all of these different model options were built with cells from the same patient. Concerning what's energy storage function, we found that the fatty acid uptake was impacted by the presence of endothelial-like barriers. And concerning what's endocrine function, we found that the adipokine release by the adipocytes was heavily impacted by the other adipose-associated cells. So here you can see 4 adipokines, which are very likely exclusively produced by the adipocytes themselves. However, in our monoculture setups, we could hardly detect these adipokines. And this finding might highlight the importance of an interplay between the different cell types for full adipose tissue function. Following up on the VATS cytokine release, in the next step, we analyzed first the basal cytokine release and release of pro-inflammatory cytokines in response to pro-inflammatory stimulation. And again, you can appreciate the contributions of the different cell types. To pave the way towards a model application in adipose immune interaction, we performed we built adipocyte macrophage co-culture systems. And in these imaging experiments, you could see that the macrophages sometimes cluster too, or sometimes even fully enwrapped around adipocytes. Moreover, since we found lipid accumulation inside of our macrophages, which can be seen here, this might indicate some lipid scavenging activity. Concerning what's or our model's capability to reflect adaptive immunity, we perfused T cells through our media channel, and we could indeed find that these T cells were recruited to the tissues in the tissue chambers. And with that, we have reached the final complexity of our WAT on chip model. However, the adipose tissue is much more heterogeneous. There are also beige adipocytes that reside among our adipose tissue. Unlike the whitekin, the beige adipocytes store their lipid contents in several smaller lipid vacuoles, and they are very rich in mitochondria. Upon a certain stimulus, beige adipocytes can become thermogenic cells. They act as a metabolic sink, and they also secrete a range of signaling molecules. If you want to work with beige adipocytes in vitro, the scarcity of this cell type in the human body can become a challenge. And therefore, usually all human in vitro research on beige adipose tissue depends on an in vitro differentiation, either through a transdifferentiation from white adipocytes or through a de novo differentiation from adipose progenitors. And the latter is what we have done in our beige adipose tissue on chip project. So we injected pre-adipocytes that were encapsulated in a hydrogel matrix and perfused them with a differentiation medium in order to obtain beige adipocytes on chip. And a very important prerequisite for the success of this differentiation is that the microfluidic platform material is non-absorbing of components in the differentiation cocktail, which is what happened in preliminary experiments with the PDMS platform I presented. So consequently, we had to develop the next generation adipose on chip platform. Design-wise, it looks a little bit different, but it shares the key features with the VAT platform. But material-wise, now here, the media layer is fabricated from a thermoplastic, which is less absorbing of small hydrophobic molecules, but at the same time also less permeant to oxygen. So we characterized the material properties of this new platform a little bit, and we could indeed confirm that the thermoplastic is less absorbing of a small fluorescent hydrophobic molecule than the PDMS version. But at the same time, fabricating the entire chip from the thermoplastic would not be favorable, which can be seen when comparing simulations of oxygen availability on chip for the version made entirely from thermoplastic versus this hybrid version. Now let's have a look at how this hybrid version performs as a stage for beige adipogenesis. We monitored the morphological changes alongside with adipogenesis, and we found that lipid droplets as well as mitochondria were increased, while at the same time F-actin and nuclei were decreased. This is how fully differentiated beige adipocytes look like on chip, and we also confirmed their maturity by staining for perylipin A, which is also for the beige adipocytes a maturity marker. Besides these morphological changes, we also had a look at the gene expression, and we found that with increasing beige adipogenesis, both general adipocyte markers as well as specific thermogenic adipocyte markers were upregulated. Concerning their functionality, our on-chip beige adipocytes responded to a better adrenergic stimulation by increasing their UCP1 production, and they were also able to take up fatty acids. And with that, I would like to thank you for your attention, and I'm looking forward to your questions.